All right, so we're going to pick up with the tour of the eukaryotic cell. Um, we mentioned that eukaryotic cells are going to have discrete organelles or small partitioned sub parts, I guess for lack of a better word, within the cell, that they're bound by internal membranes or divided by internal membranes. Plant cells are going to have a lot of the same organelles as animal cells. We'll have some variations, we'll talk about those, but for the most part, all eukaryotic cells, plant or animal, are going to have very similar organelles to them. So here's an artist rendition of an animal cell. There is no animal cell that has microvilli and a flagellum, so you're not going to see a cell that looks just exactly like this, but what the artist did was he or she just went through and drew all of the potential organelles that you could see in an animal cell. You should be familiar with a lot of these based off of your reading of the textbook. We'll go through them individually, at least the ones that I want to hold you accountable for, and then we'll do the same with the animal cell. But speaking of the animal cell, this is an animal cell, or rather the artist's rendition thereof. The two big things that you should notice that are different from the animal cell that you just saw is we have a large central vacuole, this guy right here. Okay, we have chloroplasts. Those were not in our animal cell, but I'm thinking that would be so cool if we had chloroplasts. And then the third, which is technically actually outside the cell because it's outside the plasma membrane, is the cell wall. So the cell wall, the chloroplasts, and the central vacuole are going to be the big three that we look at as being different from animal cells when we talk about plant cells. So let's just start off with the nucleus. Everybody knows about the nucleus. So the nucleus is basically the brain of the cell. It's information central. It's going to be where we find most of the cell's DNA. We will see DNA in other places, but certainly the chromosomal DNA is where we're going to find that in the nucleus. And so we're going to find the genes that one cell passes on to the next cell. The nucleus is usually a large, very obvious organelle. It's enclosed by a membrane. That membrane is called the nuclear envelope. Interesting facts about that membrane: it's a double membrane, so it's a double lipid bilayer. So if a lipid bilayer has two, so if a single lipid bilayer has two layers of lipid, then a double lipid bilayer is going to have 4 layers of lipid. The other thing that we'll notice about this nuclear envelope is it has pores or little openings to allow certain items in and out of the nucleus. The other thing we'll see tiny little ribosomes studded onto the exterior surface or attached to the exterior surface of the nuclear envelope. Keep in mind we're going to come back and we're going to look at this nuclear lamina. Okay, so we're going to look at nuclear lamina as well. This is a surface or a scanning electron micrograph of the nuclear envelope. These are pore complexes, so this is the transmission micrograph, and this is the nuclear lamina, which is a transmission electron micrograph. Remember the difference between those, scanning and transmission, that is. So some key terms we need to talk about when we talk about the nucleus. I just mentioned the pores that are going to allow entry and exit into and out of that cell. We took a peek at that nuclear lamina. That nuclear lamina is actually made up of protein, and it's what's going to give that nucleus its shape. Okay, so it's thicker. It's not rigid entirely, but it's a protein, so it's a proteinaceous base. So it's going to be a little bit thicker and a little bit more. I don't like the word rigid, but a little bit more substance to it, so that it's going to give some shape. You'll notice the little squiggle lines on this image. So the little squiggle lines right here, those are chromatin. Chromatin is actually the DNA and its associated proteins that are loosely coiling, not even coiling, they're just kind of loosely laying about inside that nuclear envelope. Kind of think of them, they're not organized at all. So think of them like a bowl of spaghetti. They're just kind of floating around in there in the spaghetti sauce, if you will, and they're just hanging out inside that nucleus. That's the chromatin. when chromatin actually starts to condense down, when we start to see it wind around those proteins, specifically the histone proteins, when we start to see it coil back on itself and wind around those histone proteins, we're going to form those really nicely discrete little units, the ones that typically get drawn as little X shapes, right? Those are called chromosomes. So DNA in its loose form, when it's just loose with its proteins, is called chromatin, and then DNA when it condenses, when that chromatin, so the DNA and the proteins, when it starts to condense and supercoil, then it's called a chromosome. One other part that I want to mention at this time, before we move on to the ribosomes, but one other thing is the nucleolus. So the nucleolus is right here. It's usually a denser, takes up more stain, more condensed area within the nucleus. So watch those spellings, nucleus versus nucleolus. So the nucleolus is a more condensed area within the nucleus. It is the site for the production of ribosomal RNA. Okay, ribosomal RNA is going to be a key component when we make up ribosomes. So ribosomal RNA plus protein is going to make up a ribosome, and that's our segue into ribosomes. So ribosomes are little protein factories. They are particles made up of ribosomal RNA and protein, like we just said. They are found in two forms, free-floating and bound. So the free-floating ribosomes are just going to be free-floating in the cytosol. Bound ribosomes are either going to be attached to the nuclear envelope or to the endoplasmic reticulum. Okay, and I want you just to think of them as little workstations. So they're like a workbench or a workstation for building a protein. They're made up of small and large subunit. We'll go into that a little bit more, how those come together when we get to translation much later in the semester. But again, these are ribosomes. They're made up of ribosomal RNA and protein, and they are the workstation or the workbench, if you will, for the production of proteins. Okay, so this is going to be a good place for us to take a break. You should at this point be able to jot down a whole lot of information about a nucleus and ribosomes. You should be, when you go back and check yourself, you should be pulling out all those bolded in key terms. You should have definitions. You should know what those words mean. You should know how they interact or fit together or the relationship between them. So I'm going to pause here and we're going to pick up with something called the endomembrane system.